Welcome to the deep dive. We spent a lot of time focusing on really high-tech solutions to complex problems, especially when it comes to things like climate change and biodiversity loss.
Right.
But sometimes, the biggest breakthroughs, they come from finding a brilliant new use for something really ancient, a natural technology.
And today we are talking about one of nature's most effective and, well, utterly ubiquitous inventions, spider silk. Or, more specifically, the entire web structure.
That's right. Today, our mission is to break down a fascinating resource article led by Gregorič and colleagues.
Yeah.
It's titled Spiderwebs as eDNA Samplers, Biodiversity Assessment Across the Tree of Life.
Yeah, and the underlying goal here, what we're diving into, is establishing that these delicate, often sticky structures are actually far more than just passive insect traps.
Much more.
They might be the next generation of powerful passive tools for sampling environmental eDNA, offering a potentially revolutionary shortcut to assessing biodiversity across the entire tree of life. We're talking animals, fungi, bacteria, all in one go.
Okay, let's unpack this. So before we dive into the silk itself, let's maybe establish the baseline problem. Why is monitoring biodiversity so difficult right now? Why do we even need a new method?
Well, the context is pretty dire, unfortunately. We're facing continuous, rapid biodiversity loss globally. But our methods for tracking it, they're kind of stuck in the past.
How so?
Traditional monitoring, it relies heavily on classical taxonomy, looking at physical traits, and that presents some huge hurdles.
Such as just the sheer volume of life we don't even know about yet.
Precisely. The vast majority of biodiversity is still undescribed by science. And even when we can identify an organism, the taxonomic expertise needed to actually do that, it's declining sharply. And often just looking at phenotypic diagnoses, what an organism looks like, it's just not enough for a definitive ID.
And there's the ethical dimension too, which feels really important here. Traditional sampling can be destructive, can't it? You often have to physically collect, maybe even kill the organism you're studying, or seriously disrupt its habitat.
Exactly right. And that's why environmental DNA, or eDNA, has become this molecular solution that's just exploded in popularity recently.
Right.
Instead of needing the organism itself, eDNA lets us gather genetic material, think shed skin cells, maybe spores, feces, directly from the environment, soil, water.
Okay.
It enables this non-invasive, high-throughput way to assess biodiversity using something called metabarcoding.
But applying that to the air, the terrestrial air environment, that sounds incredibly tricky. I mean, we can't exactly filter the whole atmosphere easily.
Sure, you can.
Which brings us to our primary hook today. Why spider webs? What's the idea there?
Okay, here's where it gets really interesting. Spiders are everywhere. I mean, literally everywhere. They're globally distributed, and their webs, whether they're tiny little things or huge constructions, they function like constant air filters. So the researchers hypothesized that these webs act as perfect bioadhesives. They trap basically everything floating in the air, not just the insects we see, but dust, pollen, fungal spores, and crucially, trace amounts of eDNA.
So they're sort of using a naturally deployed, pre-built apparatus to maybe supplement or even replace human air sampling materials. But proving this isn't just a cool theory, that required a really robust study design, I imagine. How do they move from hypothesis to proof?
They adopted a very clever two-pronged approach to bridge that knowledge gap. Part A was a highly controlled single-species test. It was designed to prove the fundamental concept. Can a spider web actually accumulate and retain the genetic traces of even a really small known target?
And they controlled this brilliantly, I thought, by choosing two really distinct web architectures, right? Different kinds of webs.
That's the key comparison. They used two native Slovenian species. First, the common garden spider, Araneus diademetus. It builds that classic, sticky, two-dimensional orb web we all recognize. Its capture threads are coated in glue, a super efficient bioadhesive.
Okay, the sticky kind.
Right. Second, the common hammock weaver, Linyphia triangularis. This one creates a more complex three-dimensional sheet web, but it contains only bare, non-sticky silk threads, no glue. Okay, so they were comparing an active sticky trap versus more of a passive, non-sticky architectural filter. That's smart.
Precisely. So for the test, they introduced two small house crickets, tiny things, just 5 milligrams of body mass each into these webs. Then they collected the webs hours later, importantly, after the crickets were removed and there were no visible remains left.
Right. No chunks of cricket.
No chunks. Then they used highly sensitive quantitative PCR, qPCR, to search specifically for cricket DNA.
Okay, that's part A, the controlled test. What about part B? the proof that these webs could handle a general survey across the whole tree of life.
Right, that was the large-scale metabarcoding test. For this, they collected webs from both of those spider species, but across 2 distinct forest climates in Slovenia, one sub-Mediterranean, one continental, and they sampled them across 2 subsequent years as well.
Okay, adding location and time variables. Exactly. They use high-throughput amplicon sequencing, targeting standard taxonomic barcodes, COI for animals, ITS for fungi, and 16S RNA for bacteria. The goal here was really to test the web's utility as general biodiversity monitoring tools. Could they pick up differences across web types, locations, and time?
Let's get to the results then. Did the tiny house cricket DNA actually show up in that super-sensitive qPCR test?
It was a resounding success, actually. They detected house cricket DNA in all 10 webs they tested. That includes both the highly sticky orb webs and the bare, non-sticky sheet webs.
Wow, okay. Both types worked for that.
Yep. This confirmed that first crucial step. Spider silk, whether it's glue-coated or not, is a powerful collector of trace genetic material, even from tiny arthropods.
That's a fantastic proof of concept.
Oh.
But the real meat of this study, I guess, is the massive biodiversity harvest from the general survey. How much life did they actually pull out of the air using these webs?
Oh, it was huge. We're talking over 11,000 unique amplicon sequence variants, or ASVs, in total. The diversity was just staggering.
11,000 from webs.
Yeah. Bacteria dominated, with 30 different phyla identified. Fungi were also massively represented, mostly the big groups, Ascomycota and Basidiomycota. And they found significant animal diversity, too, predominantly insects encompassing about 50 different families.
That level of detail from just a few strands of silk is, well, it's astonishing. But here's maybe the central counterintuitive finding, the real aha moment of the paper, wasn't it? The web structure mattered fundamentally, but the sticky webs weren't necessarily the best overall collectors for everything.
That's the finding that really forces us to rethink what a biological filter actually is. Surprisingly, the non-sticky three-dimensional sheet webs, the Lignifio ones, they accumulated a significantly higher diversity of bacterial and fungal eDNA compared to the gluey two-dimensional orb webs.
Wait, really? That completely defies common sense, doesn't it? I mean, I would assume the glue-coated web acts like flypaper, just trapping everything that hits it. If the sheet web has no sticky coating, why on earth is it better at collecting microbial and fungal DNA? What mechanism are they suggesting there?
It does seem counterintuitive at first glance, but the diversity difference was significant statistically. For instance, those non-sticky sheet webs contain genetic traces of organisms like nematodes and rotifers in about 65% of the samples. The sticky orb webs, none of them. So the suggested mechanism really comes down to air dynamics and surface area.
Okay, go on.
Think about it. The sticky orb web is basically a 2D net. It's effective for catching things that fly directly into it and get stuck immediately, like larger insects.
Yeah.
But the sheet web is this complex 3D scaffold.
Right.
It interacts with airflow very differently. It potentially creates microeddies, tiny turbulences, and might rely on static charge or maybe just simple interception by a massive complex surface area to passively filter the really tiny airborne particles, the spores, the dust, the bacteria that are just floating in the general air currents.
Huh?
So it functions less like a physical sticky trap and more like an advanced passive air purifier filter.
So the key isn't necessarily the glue for everything. For the really tiny drifting genetic particles, like the bulk of microbial and fungal eDNA, it's the architecture interacting with the air that matters more.
Exactly. That seems to be the case. And the power of these webs was further proven through the beta diversity analysis. That showed that the community composition bacteria, fungi, animals was significantly affected by the web type, the forest location, and the year of collection.
So if we connect this to the bigger picture, what you're saying is this proves that a scientist could potentially go into a specific habitat, collect just a handful of webs, and use that sample to track significant biodiversity differences across maybe different microenvironments, or even track shifts over time, like year-to-year changes in fungal populations, perhaps.
Yes, it really gives you highly localized, high-resolution data. That's something that's quite difficult to achieve with, say, broad air sampling tools that might average over a large area.
Let's talk about the broader implications, then, for conservation and human society, because this clearly goes far beyond just counting insects in a new way. Given the scary decline of global insect populations, these webs could offer a totally non-invasive platform for monitoring that crisis.
Absolutely. And the researchers actually detected several critical pollinator species, bees, flies, wasps, even endangered longhorn beetles were found in the web eDNA.
Wow.
This offers a potentially powerful solution to the growing lack of taxonomic expertise in pollinator monitoring. You don't necessarily need a specialist entomologist in the field to identify the bee. You just need the web and a sequencer back in the lab.
And presumably we could use this for early detection of invasive species too, like tracking pathogen-carrying mosquitoes or maybe newly arrived agricultural pests before they become widespread and established.
Precisely. That's a huge potential application. And the findings had massive relevance for agriculture and medicine already. They captured genetic material from taxa that are critical to human welfare. On the agricultural side, for example, they detected traces of some really serious plant pathogens.
Can you highlight maybe a couple of the most impactful ones they found, just to give a sense of it?
Sure. We'll take the fungal representative Magnaporth griseia, for instance. This is the fungus responsible for rice blast disease. It's one of the most destructive rice diseases globally, causing billions in damage every year.
Right. Huge economic impact.
Huge. Finding its eDNA in a web means you could potentially detect its airborne presence maybe months or miles before the actual disease symptoms even appear in the rice paddies. They also found things like grain rust mites, which are major crop pests, and fungal genera like Fusarium and Verticillium, notorious for causing crop rot and wilt.
That is potentially huge for food security. I mean, early detection is everything in pest management. And what about the medical implications? Were there human health relevant finds too?
Yes, medically important agents were also present in the web samples. They detected fungi known to cause respiratory problems and allergies, like Aspergillus and Stachybotrys, that's often called black mold. They also found bacteria associated with food spoilage and livestock issues, including genera like Salmonella and Clostridium. So this suggests webs could potentially be used to monitor the distribution and movement of public health threats out there in the environment without needing active air filtration devices.
This technology really sounds revolutionary, but I have to ask about the practical labor aspect. Is it really scalable? It sounds like you potentially need to collect hundreds or maybe thousands of individual, very delicate webs. How much time does it take to gather these tiny samples compared to, say, taking one big 5-gallon water sample for aquatic eDNA?
That's a really critical question, and it speaks to the sort of logistical trade-off here. While the webs themselves are passive filters, meaning they collect the data basically for free, nature does the work. The collection and processing of potentially thousands of individual sticky or fragile webs is certainly going to be more labor-intensive than, say, grabbing a single water sample.
Right. So we're kind of exchanging the expense and complexity of setting up automated air filtration equipment, maybe, for the specialized labor of skilled field collectors who know how to handle these webs carefully.
Exactly. I think that's a fair way to put it. The advantage isn't necessarily ease of collection. The advantage is the potentially unparalleled spatial precision you get, and this unique ability to monitor the airborne environment terrestrially using a natural collector. If you really need hyperlocalized data on a specific crop field or maybe a particular layer in the forest canopy, the web might be the best tool we currently have, despite the collection effort involved.
Okay, that makes sense. Let's turn now to the necessary methodological limitations and challenges the researchers themselves identified. One key issue you mentioned involved the COI marker they used for animals. It sounds like they hit a bit of a technical wall during the sequencing part.
They did, yeah. The problem was that the common COI amplicon, that's the specific genetic snippet they were trying to read, it turned out to be a bit too long for their sequencing setup. When you sequence eDNA, you often use what are called paired end reads.
Right.
That means you read the sequence from both the beginning and the end of the snippet, and then you try to stitch those two reads together in the middle to confirm the full sequence.
Sort of like reading the first few lines and the last few lines of a really long sentence and hoping they connect properly in the middle.
Yeah, precisely. And if the sentence is too long, the two reads just don't overlap enough, and you can't assemble the complete verified sequence reliably. So this meant the researchers had to essentially abandon the paired end approach for COI and rely only on single end reads. And that significantly reduces the identification confidence and power. They couldn't be quite as sure about the exact species identification for the animal DNA.
Okay, so future studies clearly need to adapt the molecular methods to fit the sample type better. Either maybe using shorter COI amplicons that can be stitched together, or perhaps deploying newer next-generation sequencing technologies that can just generate much longer single read lengths to begin with.
That's definitely the path forward on the tech side. Standardization is also crucial. Researchers really need to standardize protocols around variables like the web's environment, is it out in the sun or is it in the shade, and also the web's specific lifespan.
Right, because some get rebuilt daily, others last longer. And what about the background noise? I mean, the DNA from the spider itself, the web producer, that must contaminate the sample to some degree, right?
Oh, absolutely. It definitely contributes background eDNA.
Yeah.
They noted the critical need for laboratory controls to estimate and then computationally subtract the genetic material that naturally comes just from the web producer spider. And perhaps most importantly, especially for temporal studies, we really need to figure out the degradation rate of genetic traces on these webs. How long does DNA actually last on silk under different conditions? Without knowing that, we can't accurately pin down when a detected spore or bacteria actually landed there.
This raises a really interesting question then. Given the varying lifespans of different web types, orb webs often rebuilt daily, offering a snapshot, sheet webs lasting longer, giving maybe a cumulative picture, how might we actually integrate these webs into some kind of global standardized network? Could we get near real-time monitoring of airborne threats, tracking life at a granularity we've just never managed before?
It's a tantalizing prospect. I mean, if we can solve the methodological hurdles and the logistical training issues for collection, we suddenly have this globally distributed, self-deploying network of environmental sensors. They're available in pretty much every ecosystem on Earth, ready to collect samples 24/7. It could fundamentally change the scale of possible biodiversity tracking.
Our core insight today seems pretty clear then. Spider webs are not just biological detritus or insect traps. They are actually sophisticated, non-invasive, and remarkably spatially precise biofilters. They're tracking life across all domains, a kind of natural shortcut emerging in biodiversity science.
Yeah, they really offer a sort of molecular time capsule, capturing the genetic history of the air around them on potentially a daily or maybe a seasonal basis.
So what does this all mean? Well, for you, the learner, maybe it means recognizing that sometimes the answers to these vast scientific challenges are literally hiding in plain sight, shimmering in the morning dew.
Consider that unique temporal information provided by different webs. If a scientist collects an orb web that's rebuilt every single morning, they get a molecular snapshot of that specific 24-hour period. What new, incredibly detailed insights could scientists glean about daily microbial cycles or maybe pest activity or pollinator movements if they could read the airborne environment with that kind of granularity every single day? What will you look for the next time you see that glimmer of silk between the branches?